Good morning to everybody. As the session, all the speakers are, they don't speak Spanish, so we will do everything in English if you don't mind. So let's start with this muscle and diabetes session. Muscle is an organ that we don't usually pay too much attention, so we will try to remedy this situation with this session. So before we start, I would like to thank the speakers for the effort they made to be here with us and to share their knowledge and research. So we will begin with the first speaker, which is Dr. Lykke Sylow. She's from University of Copenhagen in Denmark. She will do it online because she's, for some maternal reasons, she's there. Okay, please, Dr. Lykke, you can begin as soon as you can.
Good morning, everybody. Thank you very much for inviting me and also for accommodating that I'm unable to travel at this time. Unfortunately, I would love to be in Spain with you. So in my talk today, I will speak about insulin resistance in skeletal muscle in type 2 diabetes and why I think this is a very important avenue to pursue. So we know that reduced insulin action is truly detrimental in several conditions, and that is because insulin is a hormone that, amongst many crucial functions, controls blood glucose. So the outcomes of insulin action, such as lowering blood glucose, is really reduced in many common conditions, including obesity and type 2 diabetes. And here there is mounting evidence that the skeletal muscle is truly an important tissue in this regard. And this has first been suggested by work by Ralph DeFronzo, showing that in normal insulin, healthy insulin sensitive subjects, the vast majority of the glucose in the body. So I don't know if you heard me at all, but I just want to thank you, the organizers, for inviting me. And I regret that I'm unable to travel at this time. And I'm here to speak about skeletal muscle insulin resistance in type 2 diabetes and why I think that this is very important and also a place where we can hopefully get some targets to treat insulin resistance. And we know that insulin is a very crucial hormone that, amongst many different functions, controls blood glucose. And these actions are reduced in several different conditions, and that includes obesity and type 2 diabetes, which is why we are here. And this heightened blood glucose in the context of insulin resistance seen in obesity and in type 2 diabetes is really due to a decreased insulin-stimulated glucose uptake by the skeletal muscle. And this was shown first by Ralph DeFronzo, showing that in healthy control subjects, the vast majority of glucose goes to the skeletal muscle in response to hyperinsulinemia. Whereas many of the different organs, glucose uptake was not influenced in type 2 diabetes, skeletal muscle was really a major site of insulin resistance, and this was where there was a lowered insulin-stimulated glucose uptake. And this is also the case in other different conditions, such as aging, and we're also starting to understand now that insulin resistance is important in cancer, but I will focus my talk on type 2 diabetes. And there really are significant gaps in our understanding of what are the molecular signals that drives insulin-stimulated glucose uptake and why are they reduced in insulin-resistant states. And understanding this better, I think, really represents a major scientific challenge for us, which is exciting, but it could also be an untapped opportunity for medical exploitation. So this is an overview, or a simple overview of the insulin signaling to GLUT4 translocation. Because when we eat a meal, glucose is increased in the blood, and then there's also an increase in insulin. And this results in this tightly regulated and complex intracellular signaling cascade that in the end results in the translocation of the GLUT4 vesicles to the plasma membrane. And this process is really required because the water-soluble glucose cannot readily enter the bilayer of the muscle membrane. So it's only when the GLUT4 vesicles sits in the membrane that glucose can enter the cell. So in this way, this signaling pathway and how this works really dictates how much, what our blood glucose levels are. And we know now that insulin resistance arises from a defect somewhere in this intracellular signaling pathway, but we're still, I think, not completely sure what exactly is going on. And classically, this molecule here, AKT2, has been seen as a major regulator of glucose uptake in skeletal muscle. But in my opinion, its prominence in this process is not entirely backed by evidence. For example, here looking at AKT knockout mice, you can see that there is a minor reduction in submaximally induced insulin-stimulated glucose uptake, whereas the response to a maximal insulin dose is actually completely maintained in these AKT knockout mice, suggesting that insulin-stimulated glucose uptake can occur even in the complete absence of AKT. So we've been wondering for some time what else is going on in this pathway that we may have missed. And we have focused very much in my team on work that was originally pioneered by Professor Amir Klipp in Toronto to try and look for alternative pathways. And here we have been interested in an emerging pathway that is controlled by the Rho GGPase RAC1. And RAC1 is a key regulator of vesicle transport in many different cells, including insulin vesicles in the beta cells, and here the GLUT4 vesicles in the muscle cells. And it does so by reorganizing the cortical actin cytoskeleton, which is then necessary for the GLUT4 vesicles to actually dock and fuse with the plasma membrane. So we have studied this, and we found that when we looked at active RAC1, and RAC1 is active when it's bound to GTP, and only then can it activate its downstream target by binding and then later autophosphorylation of this kinase PAC1 and PAC2. And this is a surrogate measure for RAC1 activity that we can measure in skeletal muscle. So we took muscle biopsies in humans that were lean or had obesity, or patients with type 2 diabetes at baseline and following a hyperinsulinemic euglycemic clamp. And here was really clear that there was sort of a stepwise decline in insulin's ability to activate RAC1-induced signaling. So this was very interesting, but we then thought, what is it actually that could be inactivating the Rho GGPases in insulin-resistant skeletal muscle of these obese people, as well as in type 2 diabetics? And two really nice studies came out from Ron Kahn's group some years ago, showing that if they took stem cells from human skeletal muscle of controls and type 2 diabetic subjects, cultured them, they could see that what differentiated the healthy and the insulin-resistant cells was very much an alteration in signaling by Rho GGPases. And this excited us because this is what we looked into, and we have now really tried to better understand what could be inhibiting these Rho GGPases, or at least RAC1, in insulin-resistant skeletal muscle. So Lisbeth Møller, who is a postdoc in my team, she investigated the RAC1 interactome in response to when RAC1 was active and when it was inactive. And what she found was that this protein, RGA, which is a gene name for GDI alpha, really heavily interacted with RAC1 when RAC1 was inactive. So it kept RAC1 in an inactive state. And we then, in collaboration with Amir Klipp's group, and this is work driven by Tim Chu, a postdoc with Amir Klipp, were able to show that when we IP'd Rho GDI alpha, so pulled down GDI alpha in the muscle, we could then see that in response to insulin, less RAC1 came down with it, indicating that insulin somehow results in the dissociation of this complex, leaving RAC1 to be activated to increase GLUT4 translocation. And we also found that rho-GDI alpha was phosphorylated in response to insulin. So we think that this phosphorylation destabilizes the complex, leaving RAC1 to be active by insulin, and that this is somehow defective in patients with type 2 diabetes. So if we quantified all of the experiments we did, we could see that with insulin, there was really a decline in the GDI alpha RAC1 interaction, and that coincided with an increased phosphorylation of rho-GDI alpha at the serine 101 site. So we now, knowing this, we really wanted to test if this holds true, if GDI really actively inhibits RAC1. And the working hypothesis was that GDI alpha, when phosphorylated, would then destabilize the complex, and thereby RAC1 could be activated by unknown mechanisms in response to insulin. So Lisbeth Møller, she downregulated GDI alpha in L6 myotubes, and you can see that she obtained a very decent knockout in these cells. And if we knock down the inhibitor of the rho-GTPases, then we would assume that we would activate the rho-GTPases. And this is exactly what we saw. So we did see that if we removed this inhibitor, the GDI alpha, we saw an increase in RAC1 activation at baseline, but also in response to insulin. And if we did the opposite, if we overexpressed this protein in the skeletal muscle GDI alpha, we saw an inhibition of insulin-induced RAC1 activation. So for us, this really suggests that we find sort of a new signaling node in response to insulin that initiated the activation of RAC1. So we next needed to, of course, prove that this also had something to do with the glucose uptake. In order to do that, we overexpressed GDI alpha using recombinant adeno-associated virus. And you can see we got 100% transfection of these muscles, leaving the contralateral leg as the mouse's own control. And what we observed was that there was a reduction in skeletal muscle glucose uptake when we overexpressed GDI alpha to inhibit RAC1. So this really suggested to us that GDI alpha is a new molecule that is important in the regulation of insulin sensitivity of the skeletal muscle. We also sought to translate these findings a little more into humans, because unfortunately mice are not an excellent model for human physiology. So we got our hands on these obese type 2 diabetic subjects, and we could see that in line with the inhibited RAC1 activity, they also seem to have a higher level of GDI alpha, which fits our hypothesis that this would then impose an inhibition of this insulin signaling pathway. So this suggests that these mechanisms could translate to the human condition as well. And when we then overexpress GDI alpha, not only in one muscle, but in all of the muscles of the entire mouse, mimicking the upregulation that we saw in the type 2 diabetic humans, we could then see that when the mice were fed a high-fat diet, that they did become even more glucose intolerant compared to the control high-fat fed mice. So these mice that had an upregulation of GDI alpha, they were more prone to developing insulin resistance and glucose intolerance. So now just going back to this slide here, I just want to provide the updated version of this now, because we now know that there's more to this signaling pathway, and I'm sure that there's much, much more than we know still. But that in order for, at baseline, rho-GDI alpha binds to RAC1, but then insulin somehow phosphorylates GDI alpha, leading to dissociation from RAC1, which then allows for the GLUT4 vesicles to go to the membrane and glucose to enter the cell. All right, so with that, I just have 3 take-home messages based on this talk and the findings we have. The first one is that I think that these molecular signals that mediates glucose uptake in skeletal muscle is really exciting and uncharted territory where there's an untapped opportunity for medical exploitation in type 2 diabetes. If we could get the glucose in there, I think that would help the condition much. And we have recently now discovered that the GDI alpha RAG1 complex is a new regulator of insulin-stimulated glucose uptake in skeletal muscle. And we know also that elevated GDI alpha protein and/or binding to RAG1 could cause insulin resistance. But of course, there's more work to be done. And I think that this is relevant in all insulin-resistant conditions, diabetes, but also aging and cancer. And this is conditions that we are exploring further in our trials. And I just want to end, of course, with the most important slide, I think, with acknowledging my team. We are the Molecular Metabolism, Cancer and Aging group in Copenhagen with all of these fantastic researchers. And we are, of course, grateful for the funding we get. And I just want to highlight Lisbeth, who has run the majority of these the studies that I've shown you. And I also want to thank you, of course, for your attention.
Thanks very much, Lykke, for sharing these very interesting results with us. If we were thinking leaving a final round of questions at the end of the talks, yep. If you could stay with us for the session, that would be great.
Yes.
Yeah, wonderful.
Of course. Thank you so much.
Thanks. So our next speaker is Dr. Johanna Lanner from Stockholm. She is Associate Professor of Physiology at the Karolinska Institute. And she will be telling us about diabetic myopathy and disease and medication and therapeutic and strategies.
Hello, everyone. I would like to start and thank the organizers for inviting me. It's a pleasure to be here. So today we will stick with skeletal muscle and we'll talk about diabetic myopathy and the impact of. There it is. I just want to see if the pointer works, if that's okay. Now it works, perfect. And I have no conflict of interest to declare. So just by taking a definition, what is diabetic myopathy? And the definition says that it refers to a broad spectrum of pathophysiological alterations to skeletal muscle in response to diabetic biomedical, hormonal, and cellular environments. So basically, a lot of things. And what this pathophysiological alteration might be, it's a big spectrum. And I'll just borrow this image actually from a review that Dr. Farup recently co-authored, where they listed, they were talking about muscle, the decrease in regeneration of muscle in the diabetic population. And they also listed several, you see here, several things that may happen in the muscle that can contribute to that the muscle doesn't work as well as it should. So what should muscle do? I mean, excuse me, skeletal muscle does a lot of things to our body, and it's essential for our health. So we should pay more attention to skeletal muscle. It's a wonderful organ. It's 50% of our body is skeletal muscle. And Lykke just told us about how it's important for energy storage, but we also know that it's essential for our mental health. It also, I mean, just for breathing, we need skeletal muscle, and of course for our movement. And to be able to move and to have a daily life and lifting things. So I will focus on the movement and the force generation as a readout for how well skeletal muscle works. And just as a summary, I mean, if we lose the ability to generate force and then get weak, that could be of several reasons. It could be that you lose the neuromuscular interaction, which you do in ALS, that you all know about, another severe disease. But also then, of course, if you lose muscle mass, it's intuitive to think that, okay, if you have less muscle mass, you also have reduced capacity to generate force. But you also have this third thing. When you look at the muscle, it looks normal, has normal size, but still it can't generate force. And that's called intrinsic muscle dysfunction. And that's, I'll tell you more about that, and that's what we will focus most on. And then we'll hear more about the sarcopenia, I think, in the next talk. So this paper came out in Diabetes in 2006 by Anderson et al. And I think this is one of the first studies where they really showed this diabetic myopathy, that is, the function was declined. It's not only that you lose the ability or have less muscle protein, but you also have reduced muscle quality. And they call then what I call muscle quality is when you normalize the muscle, how much force, so Newton meters force, and you normalize it to how much muscle mass you have. And that is decreased, kind of saying that it's not only whatever decreased in the muscle, it's not only the result of that you have less muscle, it's actually that the function in the muscle has declined. And this is in a large population of, I mean, at least about 300 diabetic patients that they measured. And they measured both the legs and the arms, and they saw the same thing. But what was also interesting in this paper is, and what the relationship, or what they stratified was for how long they had had the disease. So you saw the same thing in both men and in women. So this black bar is that if you have had the disease more than 6 years, and this with the striped one here is like if you had the disease less than 6 years. And you see that there's a trend or a correlation that the longer you had the disease, the poorer your muscle function works. And you might think that, this is not much, it's like 8 to 10% decrease in force. But that's quite a lot if you think about that. If you would be 10% weaker all the time, your life will be much harder to just walk to the bus or carry your groceries back from the store. So this kind of gives us the relationship of that somehow the disease duration seems to have a correlation to like the degree of muscle dysfunction. So the longer you've had the disease, the poorer your muscle function is. And that could, of course, be, I mean, is that just biological age that you had the disease for 6 more years, so 6 years older? Or is it the medication that pays in, or is it other comorbidities that you get along the way that also then negatively impact your muscle? And of course, I mean, most likely, it's the combination of all three. But so when it comes to medication, we know that, for example, metformin and statins can cause severe muscle problems, and metformin has shown to decrease the exercise response. And when it comes to comorbidities, like one example that I will talk more about is peripheral artery disease, that it clearly negatively impacts the muscle function. So all of this combined will, of course, lead to whatever the decline in muscle force that you have. But as you know, as a researcher, we have to take one step at a time. So we focus lately on peripheral artery disease. So peripheral artery disease, so 1 in 3 diabetics over the year of 50 exhibit peripheral artery disease. But peripheral artery disease you can also get even if you don't have diabetes. So over 200 million people worldwide are afflicted by this disease. And it starts by what you see in this image, like there's a narrowing of arteries and blockage, which of course lead to ischemia, and that will give damage to muscle. But the thing is, like, even if you open up the arteries and if you improve the vascularity, the myopathy still persists. And this says to us there's more to it than we currently know. So the fact that the muscles are still not working even if you open up this. And as you can, so what's quite fascinating is like it's also very undiagnosed, even so people can go with this a long time without knowing it. So, and also, of course, the severity ranges widely here. So what's been known is that depending on how severe this disease is that you will get more and more negative impact on muscle. So if this is a healthy muscle, this is from Clough et al., then you can tell that the more severe, going from mild to different types of moderate PAD, you see these white parts appearing in muscle, which they should not be there, but that's fibrosis. And you can also see that some of these muscles are much smaller than they are here. So you do also have an atrophy, that muscles get smaller because there's fibrosis there also that kind of makes sure that the muscles cannot have all this length. So we looked at this and we're wanting to say, okay, we have all these observations. So now we are on a quest to try and understand, so what's actually happening on the molecular level. We're not there yet, so maybe in a few years, if I can come and speak again, I hopefully have more, can tell you more about the story. But what we have some interesting findings I want to share with you. So what we did is that we took biopsies from patients with PAD. So we took biopsies either from vastus lateralis, and we call them the non-ischemic, because the insult of the occlusion of the vessels is below the knee. So we also took a biopsy from gastrocnemius in the calf muscle, and that's what the ischemia, we call that the ischemic muscle. So when we started to look, because then what's been used in the field before is that this vastus lateralis is supposed to be the healthy control because it's above the occlusion. But when we started to look at this, you see that, so the arrows here is to the blue dots. They are cell nucleuses. And the cell nucleuses in healthy muscle, they should be here, aligned by the red here is the plasma membrane. But what you could see is that you have these nucleuses both in the center of the cell, both in the ischemic and non-ischemic. The kind of says is like, okay, we have more of that muscles that is not directly affected by ischemia is actually also severely affected. So this gives us now more also, and can give us a clue why this is also important in diabetes, because you have more of a systemic effect. And this was true not only for then what we call the centralized nuclei. We have the same trend, and compared to the healthy, the fiber cross-section area is smaller with the central nuclei. It's both the non-ischemic and ischemic is higher than just healthy muscle from healthy volunteers. And we see the same pattern both for lipid accumulation, fibrosis, and the change in fiber type composition. So that says that not only this myopathy is not only local to where you have your insult, but also globally. So definitely, and then we have still, we have only imaged 7. We have more samples in the freezer. But so the trend is that also, if you have type 2 diabetes, you have a more severe kind of phenotype than with if you don't have type 2 diabetes. So this is something we're collecting more data, and this trend we will think it will be significant, but then in order to do things more mechanistic, we turn to our mouse model. So for the peripheral artery disease, we use the femoral artery ligation. So you ligate the femoral artery on 2 sides, and then you cut between. And if you then and we then combine this with diet-induced obesity. And so we treated the mice with a high-fat diet. So 60% of all the calories came from fat, compared to 10% in normal chow diet. And then we did this femoral artery ligation. And just to show you that, of course, the ones that were given the high-fat diet and the mice got obese, and they also got this reduced glucose tolerance, the same like Lykke showed you. You give a glucose bolus, and then you see how fast the body can clear it. And then just here also, this is the blood flow. The intensity is the more, and then this is, you see, directly after the femoral artery ligation, there's no blood flow to the paw, and then we use the contralateral as control. And you see then that the blood flow kind of after 4 weeks, it recovers, but not back to baseline. And also with that, we haven't seen any difference in the blood flow, how well that recovers between the obese mice and the normal mice, and that's aligned with other publications. But what the question was then, if you just have the obesity, as we have as a proxy here right now for type 2 diabetes, it's like, does that aggravate any of the muscle abnormalities? And what we looked at here, as I said, focus on force. So we take out this soleus muscle from the mice, and we put that in a force transducer, and we measure force. And the black here is how much force the muscle generates normally, and this is then normalized to the cross-sectional area of the mice. And you see that after 18 days of FEL, the force is drastically declined, but then it kind of recovers over time when the blood flow comes back. But in the mice with the obese mice, you see that first of all, they can basically not produce any force after 8 days. And also that the recovery is much slower. So there is definitely some sort of a aggravated decline in muscle function here. And we also then looked at the mitochondria, which is, as you know, the powerhouse of the muscle. And I know that this is a messy slide, but just follow me here. So basically we did a respiratory experiments on adult intact muscle, and the white and black and white here is the show diet, and then it's with a control of the FEL. And I mean, there's a decline in the FEL, but not severe. But if you look at the same, especially here in complex 1 and complex 2, parts of the mitochondria, especially after the 8 days, you have a mitochondrial decline in mitochondrial function. But this is like observation. What's actually going on? I mean, that's what we want to find out. So what we have done, and what I just show you a snippet of, is like, this is, we did bulk RNA-seq on these muscles. So basically, we just get the whole transcript we took 15 days, so the muscles had recovered halfway. And I just want you can't see the text, I know that, but you just want to see the pattern. That you can see that in the healthy ones, which is here, so the normal chow, you see that the response, so this is a volcano plot, so down here is the fold change. So to the right here is if it increases, and to the left if it decreases, and this is how significant the response is. And you can see that in the normal ones, the response is much, much higher than we can see in the high-fat diet. And if anything, there's also more decline in transcriptional changes in the obese ones. So, and maybe it's easy also in a Venn diagram to see how we compare that from this we can see that at least 900 transcription changes are unique in the obese mice. And then it's like all about like how can we find what actually matters here? And we're on the way. And I just want to show you again, just looking at the pattern. We looked at transcription factors, which are important for they are a hub that can affect many different the transcription of many genes. So I just want to highlight that you see that most of the ones, this is all the 4 groups, and you can't see what it says, but this is all the 4, and you see the pattern is red if it's increased, blue if it decreased, very similar, except for this transcription factor here, that is, it's down, this is how, with obesity is down, it's up when in normal diet, down with fat, and up with normal diet. So this transcription factor, we're definitely interested in trying to understand further. And I think this is the way how we can improve our understanding of what this diabetic myopathy is about, and especially when it comes to this severe impact of occlusion of the arteries. So with that, I just want to say that, of course, that the quality of muscle tissue in diabetic patients is definitely influenced by multifactorial factors that we need to further understand, but the disease relation, age, medication, comorbidities are definitely impacting this. And we can also, from what the data we have, is that femoral artery can work as a proxy for PAD, and we think that's important to further understand how this diabetic myopathy affects the muscle. And if you just provoke you can provoke this diabetic myopathy even further with a diet-induced obesity when working with animal models. And what we will continue with, but at least the transcription profiling highlights the changes in muscle from in PD muscle from obese mice, which are definitely different to the ones on chow diet or normal diet, but that needs a lot of further investigation. And with that, I want to thank the team, the lab, and also the collaborators and former PhD student that initiated a lot of this, and the funding agencies. Thank you very much.
Thanks very much, Johanna. We take note that we should invite you in some time so we get the extra results. And our next speaker is John Farup. He's Associate Professor at the Department of Biomedicine in Aarhus University in Copenhagen. And he will tell us about the atrophy and degeneration of the muscle, the skeletal muscle, and its interactions.
So thank you also for the kind invitation, and just a minor correction. So I'm not from Copenhagen. I'm from the second largest city in Denmark, so that's Aarhus. I know it's more difficult to pronounce, but hopefully we can teach you that at some point. So my talk for you today will focus on skeletal muscle degeneration in type 2 diabetes, and a couple of different aspects of this that we've been exploring in the past couple of years. And so I have nothing to disclose, and I just want to start out by acknowledging the people involved in these projects, in particular Andreas Møller, who did a lot of the autophagy work that I'll show you, and of course also the funding agencies. So, with that, as already highlighted by my colleagues today, skeletal muscle is a key tissue when we talk about the development, but also the prognosis of living with type 2 diabetes. And this was elegantly demonstrated more than 30 years ago by Ron Kahn's group, in which they showed that even decades before the onset of type 2 diabetes, you can detect changes in the insulin sensitivity of skeletal muscle, allowing skeletal muscle to predict the later onset of type 2 diabetes. Now, of course, this has spurred a great deal of research into understanding how we can prevent this, or at least delay the onset of type 2 diabetes. So what has been less well understood is how living with type 2 diabetes for decades impacts the skeletal muscle and transforms the skeletal muscle. And we've been interested in trying to understand what happens in the skeletal muscle when you have type 2 diabetes. And so, as already nicely highlighted by Dr. Lanner, we know now that when you have type 2 diabetes, the skeletal muscle transforms in many different ways, some of them on a gross morphological scale where we see fibrofatty infiltration, we see markers of muscle damage, and of course also we see mitochondrial dysfunction, along with a host of other changes in the muscle in this process. So to dig further into this, so on a gross morphological scale, you can easily visualize this, hopefully, up from these MRI images of a healthy control muscle and type 2 diabetic patients on the right, where you see this massive deposition of non-contractile tissue, which we typically associate with neuromuscular diseases, but this is clearly also evident in type 2 diabetic patients. So to explore this further, a few years back, we collected tissue from healthy but obese control subjects and heavily insulin-resistant type 2 diabetic patients, and these patients require daily insulin to maintain somewhat normal glucose, blood glucose levels. And so we looked at the transcriptome in these patients, and what really struck us was two main things. First of all, the downregulation of genes related to oxidative phosphorylation and markers of mitochondrial dysfunction, but also a marked enrichment in genes related to muscle degeneration and muscle regeneration. So one of these was embryonic myosin heavy chain, which we again typically associate with neuromuscular diseases or muscle regeneration, but this was highly enriched in these patients. And this actually struck us to be a fairly similar phenotype to what we see when we have a defect in autophagy in skeletal muscle. And I'll just come back to autophagy in a second, but this is just to show you work from Marco Sandri's group where they've knocked out HEG7, a key protein in autophagy, specifically in skeletal muscle. And what you see here is sarcomere disorganization and also swollen dysfunctional mitochondria, again resembling a dystrophic phenotype. So we thought that this could be worth exploring in type 2 diabetic patients. So what is autophagy? Autophagy is a highly conserved process allowing the cell to degrade either whole organelles, proteins, lipids, and eventually reutilize these in situations, for instance, of starvation, allowing the cell to provide substrates for energy metabolism or protein synthesis. So some of the key players involved in this is the activation of OLK1 and the OLK1 complex, which allows can I point with this? Yeah, which allows the class 3 PI3 complex to translocate to the building autophagosome and provide phospholipids to encapsulate either proteins or organelles such as mitochondria that have been targeted for degradation. Along with this, we have this E3-like conjugation system that provides the lipidation of this protein LC3 into LC3B2, which can then associate with the membrane of the building autophagosome. So LC3B2 is utilized quite frequently as a marker of autophagosomal content of the cell. And what is regulating autophagy? Well, the nutrient status of the cell is a key driver of autophagy. So when you have starvation, you will activate AMPK, and this will initiate non-transcriptional activation of autophagy. And conversely, if you have excess nutrients or insulin, this will inhibit autophagy. So based on our RNA-seq data and the fact that insulin, an amino acid, is a potent stimulator or inhibitor, of autophagy, we reasoned that this could be influencing the ability of the muscle cells to initiate autophagy. So we determined the protein content in these severely type 2 diabetic patients for LCB2, and as you'll notice here, we see a decrease in the content of LCB2. And we also see a decrease in P62, which is a key protein involved in specific targeting of proteins for degradation in the autophagy process, along with a decrease also in HEG5. So we interpret this as an inhibition of autophagy in this situation. Now, of course, I should mention that this is a difficult interpretation in the sense that we're looking at a flux, and if this was a cell model or if this was an animal model, we would utilize a lysosomal inhibitor to prevent the lysosome from fusing with the autophagosome and thereby actually determine if we had changes in flux. And this is, of course, difficult to do in humans. So, but we have other indicators that autophagy is indeed inhibited. So what we also noted was that there was an increased phosphorylation of the transcription factor FOXO3A, and this inhibits it from translocating to the nucleus and transcribe genes involved in the autophagy process. And along with that, we also found indicators of increased protein synthesis through the mTOR pathway, here quantified by an increase in phospho-4AB binding protein 1. This is a decrease here, but that's non-phospho, so this is an increase in the phosphorylated form, indicating that protein synthesis is also activated in this process, which would again serve to inhibit autophagy. And this is also supported by data from some of our colleagues at University of Southern Denmark. Here you see data from lean, obese, and type 2 diabetic patients during the basal state and in hyperinsulinemia. And hyperinsulinemia efficiently inhibits autophagy, but it also does so in type 2 diabetic patients. So we think that although you have type 2 diabetes and you're insulin resistant, you have an impaired ability to take up glucose, part of the insulin effect is still retained. So insulin can still inhibit autophagy and to some extent also stimulate protein synthesis. And this puts us in a situation where we have extended periods of time with hyperinsulinemia and potentially also increased amino acids, activating mTOR and inhibiting autophagy in these patients. And this puts these patients at a spectrum of autophagy that is insufficient to degrade the organelles that have been damaged and eventually causes this, we believe, this change in morphology that we see in these type 2 diabetic patients. So, another aspect that we noted from these RNA-seq data was a marked enrichment in genes related to extracellular matrix formation, which is also a hallmark of muscle degeneration. So to look further into what is going on here, so we also quantified the protein content of various collagens, and indeed this is increased in type 2 diabetic patients. So to look further into this, we utilized single-cell RNA-seq to figure out which cell population could be driving this fibrogenic remodeling of the cell. And here we found this major population that we termed fibroadipogenic progenitors. And so to understand if these could be driving this process, we developed a strategy to isolate the cells. And here you see them to the right, isolated after 3 days, and they're uniformly positive for this mesenchymal stem cell marker, PDGF receptor alpha. And so when we push these cells into either a fibrogenic or an adipogenic lineage, these are highly capable of forming both fibroblasts and adipocytes, and is really the only cell type that we've been able to isolate from skeletal muscle that can do this. So we think that these are responsible for the fibrofatty infiltration in skeletal muscle. And so to understand a little bit better if this population of FAPs is one major population or several different subsets, we utilized the single-cell data that we had and performed a pseudotime trajectory analysis. And this allows you to predict if there are stem or progenitor cells within the population that could give rise to the remaining population. And when we do this, we find an origin of the cell population down here in the green population, population 2, and this was also enriched in another mesenchymal marker, Phi1 or CD90. And when we isolate FAPs from human skeletal muscle, we can indeed identify both a CD90 positive and a CD90 negative population. And when we then look at their behavior, indeed the CD90 positive cells are much more capable of forming clones and entering the cell cycle, suggesting to us that these are indeed a stem or progenitor cell population within the FAP population. What we also noted about these cells was that they had a marked increase in their capacity to produce extracellular matrix. So here you see collagen 1 and collagen 3 markedly enriched in the CD90 positive population at the mRNA level. This also is true when you look at protein expression. So based on this, we hypothesized that this could be the cell fraction that is really driving the fibrogenic remodeling in these type 2 diabetic muscles. And indeed, when we look at type 2 diabetic muscles, we do see an increase specifically in this population. And even when we look specifically at this population, the cells that derive from type 2 diabetic patients are still more capable of entering the cell cycle, suggesting to us that they have really been primed in the environment to divide. And the million-dollar question is, of course, what is driving this? And this could, of course, be many things in this disease, but insulin is, of course, a usual suspect in this situation, and it is a potent driver of activating these cells. So again, we think that these extended periods of hyperinsulinemia or even exogenous insulin could be transforming the muscle in this way. And so just very briefly about the fatty infiltration part of it, which is also clearly evident in type 2 diabetic muscle. So we recently looked a little further into this together with a good colleague from Zurich, Catherine de Brock. And here you also see single-cell RNA-seq data from human FAPs from skeletal muscle. And what you'll note here is the blue population that is clearly enriched in genes related to adipogenesis. And so we sorted out these blue cells from human skeletal muscle, and when we do this, we can indeed confirm that these are markedly more capable of generating adipocytes compared to the remaining population. And when we then look in heavily fatty infiltrated skeletal muscle, we actually see a decrease in the content of these blue cells, suggesting to us that in this situation, they're really primed to enter differentiation and spontaneously differentiate into adipocytes in skeletal muscle. So finally, why do we think this differentiation of the FAPs, this fibrofatty infiltration, is a challenge? Well, we know that this is associated with a range of different diseases and poor prognosis. So this could be due to many different things. We think that one of the reasons is that these cells are supposed to have homeostatic functions within the muscle, and when they differentiate, you lose this function. And part of these functions, we believe, are actually to enhance the ability of the skeletal muscle to take up glucose. So what you see here is an experiment where we incubate myotubes with conditioned media from FAPs, and you see that the response to insulin is enhanced in this situation. So you can imagine if you lose these cells, then you will also potentially impair insulin-stimulated glucose uptake. So with that, I just want to thank you for your time, and I look forward to questions.
Since, for a change, all three presenters have kept on time, and we've had some technical struggle at the beginning, I think it would be only fair if we can steal 5 minutes to the coffee break and get some questions from the audience. Other questions from the audience?
Sorry, here, are you hearing me here? Yeah, okay. My question is regarding if you have any, for the last presentation, Dr. Farup, you have any experimental evidence regarding the effect of GLP-1 receptor agonists in this turnover of the muscle on an experimental point of view, or I can extend to the other member of the table this question.
Is it on? Oh, it is.
But based in the fact that it's an increasing of uptake of glucose, of course.
So just to understand, the question was regarding GLP-1 agonists in relation to the FAPs?
In relation of the metabolism of the muscle of any of your findings.
Yeah, it's a good question. The quick answer is we don't know. So, of course, to the extent that the GLP-1 agonist delivers a substantial weight loss, this will, of course, influence and also, to some extent, reverses the insulin resistant state and lowers insulin. This should ameliorate some of the effects that we see. However, GLP-1 agonist also seems to be to some extent a little harsh on muscle, so they seem to be causing also some muscle wasting, which is of course expected with a weight loss, but maybe even more muscle loss than you see with a normal diet-induced weight loss. So this is definitely something that we would like to explore, but we don't at this point have any answers to it.
Okay.
I think my co-chair has some
Yeah,
questions.
yes. I have a question from Johanna. Well, two, but I will do it only one, which is there any information about the role of myokines in this intrinsic muscle dysfunction?
Yeah, no, we haven't quantified any of the myokines. We are now doing a O-link, like a proteomics analysis, and hopefully that could give some indication if there's enhanced or declined myokine release from these muscles.
Okay, thanks a lot. In addition, I have a question for Lykke Sylow, so which is about as far as I know, there are some proteins which are involved in GLUT4 docking to the membrane. So is there any evidence if this is like, I don't know if that are the same one on the snares or any other ones, but is there any evidence about any dysfunction in this docking?
Yeah, it's a great question. The evidence that there is that the snares, the V and the T snares, that if you knock them out or if you knock down some of the proteins that regulate this process, there is a reduction in GLUT4 translocation. But because we don't know how their activity is regulated, and it's likely regulated by localization, which is what it's difficult to image, I don't think we have evidence to suggest that there are dysfunctions in type 2 diabetes. I simply think that because it's not a simple phosphorylation site or something like that, it's difficult to measure, so we don't know yet.
Okay, thanks. I made the question because in beta cells and in neurons, and that diabetes affects these NERS proteins.
Yeah.
Okay, we can take one more question.
So, good morning. Thank you very much to all the panel, to this Scandinavian panel. So, so I don't know who of you can answer my question, because it's a clinical question. I am an endocrinologist, and I'm also interested in also the muscle metabolism. So my question is regarding, as you know, SGLT2 inhibitors have been extensively used in type 2 diabetes because of, they have many things in prevention, cardiorenal complications in people with type 2 diabetes. Even these agents are also used in people without type 2 diabetes. But we observe that some patients using SGLT2 inhibitors, they have also sarcopenia. So they lost a lot of muscle. Do you have, and probably many of these are people who are insulin deficient or are not producing enough insulin. Do you have any explanation to explain how SGLT2 inhibitors or the use of SGLT2 inhibitors are also associated with loss of muscle, skeletal muscle in some patients? Do you have any hypothesis, maybe any hypothesis why is, because this is true, there is a lot of publication regarding sarcopenia and the use of SGLT2 inhibitors.
Yeah.
I mean, straight off my head, I do not know, but yeah, I don't.
No, I mean, usually anything that is providing a better situation for the kidney is also good for the muscle. So it's, I know that I believe there are also positive effect on fibrotic remodeling in the heart with these inhibitors, and that, to my knowledge, seem also to be at least partly independent of the glucose lowering effect. I mean, we know that the FAPs that we work with are present in more or less every tissue of the body, maybe except from the brain, and in particular also in the heart. And we know that those cells are important for maintaining muscle, at least from mouse models. So I could speculate that there would be some level of interaction there, but it would be purely speculation. Yeah.
Thank you.
If I can just add to that, because we know that they lower insulin levels, and insulin is actually a really important growth hormone for muscle tissue. So if, like, in the old days when we didn't have insulin, this was what people died of, was accelerated loss of muscle mass. So perhaps it's just because of the lowering of circulating insulin that then causes sarcopenia.
Thank you.
Okay, I'm afraid we have to stop here. Thank you very much to the three presenters for the efforts to be here with us today, and to the audience. Thank you.
Thank you.